right? So basically, people who are not here, they'll be able to watch the video and be clarified on what's happening with the exams, right? So about the fifth exam, it's not cumulative, right? It's on the day of the final, and it's going to be on material that is presented in class, starting from like today, right? From now, from this class, until whatever we cover, okay? Now, the final cumulative exam is optional. In case you have 5 grades, in case you have the 5 exams and you're happy with your grades, you don't have to take the cumulative exam, right? In case you don't have 5 grades and you want to take the cumulative exam, so you have to take it, otherwise you are in trouble, right? Now, I have been very, very flexible in terms of allowing you to take exams, right? Because a lot of people got sick and life happens and things happen, and believe me, I know, all right? So I have been giving you options to take the exam online, to take the exam at the student center, and etc. So hopefully you do have your 5 grades and you have the option to take the cumulative exam. Right? Now, that cumulative exam is going to be on the same day as your 5th exam, okay? Now, we are giving a big block of time for that day of the exam. So you're going to have your 5th exam that is not cumulative. As soon as you hand it in, you can pick up your cumulative exam if you want to take it, okay? Now, for the cumulative exam, I said that you can have your own cheat sheet, right? Everybody remembers that, right? Make sure you prepare that cheat sheet like really well. Only one sheet. And yes, you can write on both sides. Regular size sheet, like the regular size. Don't come here with a poster, okay? Regular page, right? Or smaller, yes. You can type, you can do whatever you want, it's your page. Okay, you can write on both sides, you can type, you can use different colors. You cannot share it with your friends. Okay, it's your own cheat sheet. Okay, so any questions about that? No, we all clear? Yeah. You mean for the final, for the cumulative exam? It's from the beginning of the class. It's everything. Everything that we covered. Okay? Okay. Now, regarding the my lab and mastering, okay, I've also been very, very flexible and lenient with the deadlines. I'm not even considering deadlines. So I'll give everybody the chance to complete those assignments, right? I've been getting a lot of emails and I have not been able to keep up with those emails. So I'm going to ask Parsa, please, if you can open the deadline for those assignments without penalty until the day of the final. That's fine with me. Okay, so you can catch up with that. Now, concerning chapter 10, which we're going to cover a little bit in class today, that, yes, the Malawi Mastering counts for your grade for the chapter 10, right? But of course, I'm not going to go into all of the details that are on the Malawi Mastering because I'm sure you also had that in your lab, right? You had a lot of these muscles and things in the lab. So we're not going to go into a lot of details in that. So what we cover in class is specifically on some muscles that I just put up in the PowerPoint, and I'm going to emphasize here. Those are the ones that you have to know. Okay. So after we finish this whole muscle units, actually this one should be saying 9 and 10. Okay. So after we finish all these muscle units, then we're going to go straight into the nervous system, right? Chapter 11. And of course, there is a lot to cover in nervous system, so that we're not going to be able to cover everything, but we as we usually do, we're going to focus on the most important points. All right, so we do have several questions for extra credit, and that's the other thing. I'm still getting emails about the extra credits. Those are extra. You're going to get them added to your points at the end of the term, right? So whatever extra credits you accumulated in class, those are going to be added to your points. And yes, you can bring up your grade a lot just by extra credits. And yes, still submit those questions, right? Because you do get extra credits for that too. Are we all good with that? All clear? Yeah. Oh, great. Okay. All right. So we stopped here last time, and I had stopped on this slide saying that the difference in muscles between, between the sexes, between males and females. So here, females have an average 36% body mass that is composed of muscle, but then of course that varies a lot. This is just in general, right? In comparison to males, they usually have about 42% of body mass that is muscle. But again, if you look at somebody like my friend Lucas, I think he has 90% of his body mass in muscle, right? Because he's all muscle. I mean, you can count, you can contour all the muscles that you can possibly think of, even ones that you didn't think existed. He has them. Okay. Now, it's really cute because they just had their second baby girl that last week, and she's so adorable. I am so glad I'm past my baby factory, manufacturing time, because you see those little cute things, and you're like, oh, I want one too. No, I'm done. Okay, so coming back to the muscles, muscles. So about 36% for body mass for women and about 42 for men. Now, men usually you have more more muscle mass because of hormones that act on building up muscles. Like for example, testosterone. Right? There are other anabolic hormones that can also help you build up muscle mass, right? Remember that your muscle cell number is not going to increase, but the myofibrils within a cell, those can increase like a lot, right? So testosterone is one of the hormones that influence muscle mass. Can you tell me another type of anabolic compound that can influence muscle mass buildup? No, that's for muscle function. Creatine phosphate. Remember function of creatine phosphate? Yeah. To do that for substrate level phosphorylation of ATP, right? ADP to ATP. Everybody heard of steroids? Yeah, those are banned at the Olympics and things like that, right? Steroids, like for example, inhalers that you take for bronchodilators. So people with asthma are always inhaling, taking steroids because they have to, right? But those can also build up muscle mass if you also exercise, right? So anabolic types of hormones and chemicals can definitely build up on muscle mass, including cortisone. Yeah. You see, I don't know how the rules work with those official sports, but I think that if they have prescription and history of asthma, if you think about it, a person with asthma wouldn't really qualify physically for the Olympic Games. You're competing against athletes all over the world and in the best shape. So I really doubt if somebody with asthma would make it that far. But in case they do, I think they will have plenty of medical explanation and exception to take their steroids and things like that. Yeah. But yeah, I mean, it's so competitive, it's insane. It's very insane. Even like bend your muscles. I get very, very excited with the sports thing. Yeah. Yeah, and we are like training for the winter show. That's insane. And avoid sarcopenia, but up to a certain extent. Because if you just let your muscles waste to the point that they get replaced by fibers that are, not muscle fibers, right? Then you get you get to a point of no return, that you cannot really recover your muscle after that point. Okay, so here, this is a point on homeostatic imbalances, right? So muscles need exercise to remain healthy. And if you don't use them, you get atrophy, that is degeneration, right? Loss of muscle mass. If you have immobilization of a limb, for example, like you break your arm and you put it in a cast, that also causes atrophy, right? Loss of neuronal stimulation that causes atrophy. Now, this is the thing that, you know, why the muscle strength can decline like up to 5% a day without usage, right? I mean, it takes a long time to build up that muscle mass, and then like, why 5% a day? That's a lot. Okay. Now, in case you do have atrophy and loss of muscle mass, you have buildup of fibrous connective tissue that can replace that muscle mass. And that's when you will reach this point of no return, because no matter how much you try to rehabilitate that muscle, there's no muscle anymore there to rehabilitate. You cannot exercise fibrous connective tissue, right? It's not muscle. Okay. All right. Now we get to the smooth muscle, and that's the part that we're not very familiar with, and a lot of people haven't even seen what a smooth muscle looks like. We just know that they exist, right? So this is the part that I really want you to understand how the muscle, smooth muscle, looks and how it works and where you find them, okay? So here, basically found in walls of most of the hollow organs, right? So here you have respiratory, digestive, urinary, whatever is hollow organ, you're gonna find the smooth muscle, except in your heart. What kind of muscles you have in the heart? Cardiac muscles. Are they the same as skeletal muscles? No. Of course, you're going to look a lot into cardiac muscle when you get to the AP2, that you're really going to look into cardiovascular system and going to go into a lot of details in that, but no, just the basic differences between these types of muscles, like for now, okay? So here, not found in the heart. Here, most smooth muscle is organized into sheets, right? And they are these very tightly packed fibers. So you also call fiber for a smooth muscle cell, right? Same thing. Okay, so here, I really like this video. So I basically borrowed the whole idea of the video for this class because there was a very quick everything that I like to know about this, right? So it's not like I just went on YouTube and I picked up a video. I watch a lot of videos and I do a lot of selections to really make sure that they are giving you the right information. Whatever I put in here, you can trust, okay? So I really like this video. So they gave this mnemonic to remember where you find smooth muscles, okay? So this guy gave you this mnemonic stove, right? So for most smooth muscle location. So here you have S for the skin. Like skin, yeah, you do have smooth muscle in your skin. That's your erector pili muscle. Remember those? They give you goosebumps, right? And really don't have any other use, right? Okay, so those are the smooth muscle in the skin. It's like it's right there, okay? Erector pili muscle. And this is showing a person with really a lot of goosebumps or feeling really cold. So the T is for tracts in the reproductive, respiratory, and urinary system, okay? So those are also where you find smooth muscles. The O is for hollow organs. So O for hollow, it kind of looks like O, all right? And then you have V for vessels. Those are arteries. Now your veins do not have smooth muscles that contract. Okay, so your arteries do, but not your veins. Okay. And E is for the eyes. Now, of course, not the muscles that are moving your eyes around, but specific muscles in the eyes, like when you contract the iris, right? Dilation or contraction of the iris, as well as the lens. When you are focusing, like either close or far, right? You have some contraction of the lens that is coordinated by smooth muscle. So shape of the smooth muscle. This is really cute. It looks like a football, American football, right? It has to be American football because everywhere in the world, football is not football, it's soccer. You know that, right? So we're talking about this soccer. No, not soccer, this football. It's kind of like long, right? They call it fusiform. And it has one nucleus in the center, is not multinucleated like you see with the skeletal muscle. Right? All right, so comparison of the muscles here. When you look at skeletal muscle, we know what they look like, multinucleated. So the fibers have a lot of nuclei, and then you have those striations, right? So this is like long and cylindrical. Now, when you look at cardiac muscle, you also have striations, but the muscle fiber is basically has one nucleus. Sometimes you have two, but mainly one, and is not long and cylindrical and parallel like the skeletal muscle. Those are basically form these branches. Okay, so this is the main way you can distinguish the cardiac muscle from the skeletal muscle. So this is as much as in detail as I want you to know on how to distinguish cardiac from skeletal muscle. All right, very, very simple. So when you look at the smooth muscle, you'll see that they have this kind of long fusiform shape, and they are nucleated, and you're not going to see striations. That's the main difference, right? No striations. No. So here, like skeletal and cardiac muscles, we do have those smooth muscle cells. They're separated by connective tissue. But in the smooth muscle, you only have endomysium, right? Within the muscle, endomysium, right there. You don't have the other connective tissues with the smooth muscle, only the endomysium. So smooth muscle has only one nucleus, they're usually at the center. Again, if you look at skeletal muscle, you're going to have multinucleated. You look at cardiac muscle, you're going to have one or two per cell. Yeah. Here, starting with a lot of details on this smooth muscle and how they are layered and how they work, right? So most of your organs, of your internal organs, have two layers of this smooth muscle, but they're not just on top of each other. They are perpendicular to each other. So the fibers are oriented at right angles or 90 degrees, okay? So you have one longitudinal fiber. Now the longitudinal means that it's parallel to the long axis of the organ, okay? So the longitudinal one, as you see here, okay, where is the longitudinal one? This one here. The fibers are running along, like parallel with the axis of this end. Okay, so let's say that's intestine, right? So one axis, parallel fibers going this way, and then you have this circular layer. This is circular layer. It goes 90 degrees in relation to your center axis. Okay, so that goes, the circular layer, of course, goes around the circumference, right? Now, when you look at the function of these muscles, if you contract these muscles that are longitudinal, you're going to shrink the organ, right? You're going to shorten the organ. If you contract the muscles that are in the circumference, you're going to squeeze the organ or constrict it. Okay, so you put them together and you get mixing and squeezing and pushing, and you call that peristalsis, right? Everybody's familiar with peristalsis? All right, cool. Okay. So here, this is from that video, right? So smooth muscles have layers that work together to propel food, to mix and push, right? That's your peristalsis. Now, the stomach has actually a third layer of these smooth muscles, right? So this is an oblique layer that also helps to, break down, digest the food. So more on these smooth muscles here. They are very much different from your skeletal muscle, okay? So they do have this spindle-shaped fibers, actually fiber, like this football-shaped fibers, right? Now, we mentioned no striation, endomysium, right? You have the endomysium. Now, they don't have that neuromuscular junction as we're seeing with the skeletal muscle. Instead, they have these things these little bulbs here, they are called varicosities. Okay, so these are bulbous swelling of this nerve fiber that is going to tell the muscle something to do something. Okay, so these are little bulbs. These are not found at the end of the nerve like you have with the synapse, right, with the neuromuscular junction. So those are called varicosities. Now, this is a main difference, right? It is red, it is bold, it is underlined. So do you have to know it? Yes, somebody write me a question about it. Thank you. Okay. Now, another thing that is very important that is not written here, but I absolutely want you to know is that the neurotransmitter for skeletal muscle is what? Acetylcholine, right? What are the neurotransmitters for smooth muscle? A lot of them, okay? Usually not acetylcholine, but you do have a lot of neurotransmitters, not only neurotransmitters, but also hormones can activate these muscles. Yeah. Yes, gap junction is very, very important. We're getting there. Yeah, you're a bit ahead of me here. Yeah, we're getting there, but it's very important. Keep gap junctions in your mind, okay? So varicosities, major difference, okay, between this and the skeletal muscle. And also the neurotransmitter that goes to activate the muscles that come through these varicosities, okay? Those are different than acetylcholine. There are many other neurotransmitters that can work that. Okay. Okay. So another very, very important difference. All these muscles are innervated by autonomic nervous system. That means involuntary control. Okay. Involuntary control. You just don't tell your intestine like squeeze. I mean, you can tell it, but it's not going to listen, right? So it's involuntary control. Cardiac muscle is also mainly under involuntary control. So here, we already seen that. So unlike cardiac and skeletal muscles, now your smooth muscles do not contain these myofibrils or sarcomeres. Okay, there are no sarcomeres. You don't have sarcomeres there. That's why you don't see striation in smooth muscle. Okay, so this is your skeletal muscle showing the sarcomere, right? Now with the smooth muscle is different. No striations, no sarcomeres. Okay. Now what you do have is a different system of contraction, but it also works by sliding filament. So how is it that you have here? First thing that you have to think about is how is it the muscle is going to contract. Okay, so the contraction, there are several similarities with skeletal muscle, but there are several differences too. Okay, first thing, you are going to need calcium, right? You're going to need to have calcium getting inside of the cell to activate the contraction, but this calcium is not coming from the sarcoplasmic reticulum like you have in skeletal muscle. No. Smooth muscle also has sarcoplasmic reticulum, but is not relying on it for calcium. The calcium is going to come from the outside, okay, from the extracellular space, okay, for the smooth muscle contraction. So the smooth muscle is going to have this little indentation in the sarcolemma. That's your membrane, same way as you have in the other muscle, right? So you have the sarcolemma membrane here. You have this little indentation that is called caveola, right? So here you're going to have calcium that is on the outside, that once you send a signal, or actually your autonomic nervous system sends a signal for this muscle to contract, you're going to open here voltage-gated calcium channels that allow the calcium to come in from the outside, okay, and then go to contract your fiber, to trigger contraction for the fiber. Okay? All right. So those pouch-like foldings are called caveola. Okay? There's no T-tubules there. Okay? No T-tubules. So what else you have here? You do have this contraction that are happening with sliding filaments, but it's just not the same as these smooth muscles. You don't have those striations here, right? So what do you have here? You have your thick filaments, okay? You also have some thin filaments, okay? And then you have this, of course, the nucleus here. You have these intermediate filaments that they form this net-like structure around and all inside. Here is only showing around, but imagine this, this is 2D, but imagine if this is in 3D, okay? So you have this all going into the muscle here. You have this net type of looking intermediate filaments. Those do not contract, okay, but they serve as holding places for the other filaments to contract. So whenever you have here a crossing of these intermediate filaments, you're going to find these spaces, these structures that you call dense bodies. Okay, so the dense bodies are going to hold here your thin filaments that then are going to contact with your thin filaments right there. So between dense bodies, you're going to have thin and thick filaments. Okay. Another very important point is that you're going to have a lot of gap junctions all around the surface of your smooth muscle. So these gap junctions are going to be very, very important for communication of contraction between the muscles, which you're going to see in a minute here. Okay. So here you do have your thick filament that is myosin, still myosin. It's a little different than the other myosin, but it's still called myosin. And you also have your thin filament that is your actin. So here, you have here those intermediate filaments that we hadn't seen before, right? So those don't contract. They're like the support for the other filaments. And then you have these dense bodies. You have the gap junctions, right? So here, the dense bodies attach to the sarcolemma. So that's in the cell membrane, right? Now, the function of those dense bodies is equivalent to or analogous to the function of the Z discs in the sarcomere. Okay, so these are about the same function. Now, keep in mind you do have a lot of these gap junctions, and those are really, really important for transmitting the signals for contractions between those muscles, the muscle fibers. Okay. Okay, so here, these muscle fibers usually they are connected by electricity, or they are electrically connected by those gap junctions. So they can send signals to each other via these gap junctions. Okay? When they send a signal, what kind of signal they're going to send? Electrical signal. That's a depolarization signal for contraction. Okay? So they're going to spread the signal from cell to cell because the signal now is able to jump through this gap junction to get to the next cell. If you don't have gap junction, then the signal is insulated to the inside of the membrane. Now, having gap junction, the signal can go through that hole and connect to the next cell. Okay, so your next cell is going to be like, and that cell is connected by this gap junction here. There is a hole in the middle, okay? So now your depolarization signal is able to leak through the other cell. Okay. Now, when you look at those sliding filaments, right? So here you have thick filaments that are basically not the same as you have in the skeletal muscle, right? So those are fewer and they have a lot of more myosin heads that is not only at the end of the myosin, but is throughout the myosin. So it's not going to have one thick filament and a myosin over there. You're going to have a thick filament and myosin heads like throughout that thick filament, okay? So the ratio here also from thick to thin filament is like 1 to 13, which you find here is a lot less than if you find it in skeletal muscle. There's like a 1 to 2 ratio. Okay, so you have a lot less thick filament in comparison. Okay. Again, you do have heads along the entire length of that thick filament. Okay, now this is another important point here. Okay? When you look at that sliding filament way of contracting, remember with the skeletal muscle you have troponin and tropomyosin, right? So here you don't have troponin. Remember the function of troponin? Yeah, troponin is the anchor, right? The anchor on the thin filament that when it senses calcium, it changes configuration and exposes the myosin binding site, right? So here there is no troponin, okay? Now what's going to happen here, you do have another protein that takes the place of troponin. This one is called calmodulin, okay? So smooth muscle has calmodulin. It does not have troponin. Calmodulin is going to sense different concentrations of calcium, right? Calmodulin is going to bind calcium. Now, smooth muscle also has tropomyosin. Okay, so it doesn't have troponin, but it does have a tropomyosin. So it has calmodulin and tropomyosin. Let's see how it's working here. Okay. Now, those intermediate filaments, they are made up of different proteins. Okay, so the desmin and vimentin. I don't really care that you remember the name of those, but just remember that you do have these intermediate filaments. That's important part here. Okay. Okay, we did mention the dense bodies. Okay. All right, so here we're going to start looking at the contraction mechanism of these thick and thin filaments that are between those dense bodies. Okay, so you can imagine if they're between these dense bodies, when they contract, they're going to squish this net together, so the cell ends up looking like it has little bulges. It looks like that, like poofy or bulgy. Okay. Okay. So efficiency, energy efficiency of smooth muscle, they're usually very, very efficient, okay? So there's lower to contract and there's lower to relax, okay? Now, once they contract, they can keep that contraction for a longer period of time without using a lot of energy. Now, the ATPases that are governing that contraction, they are also slow, okay? So think in terms of sloth movement, okay? Slow things. Now, those myofilaments, right, those actin and myosin, the sliding filaments, when they contract, sometimes they might just latch together. They might stay locked in place without really using energy. So they stay contracted without really using energy. And that's a good thing because there are some muscles that they have to stay contracted. And it's a good thing if they don't get tired too, right? So you don't develop fatigue and still the muscles are contracted. So that is called smooth muscle tone. Can you tell me one area in which you like to have a smooth muscle tone of this kind? Intestine, yeah. Specifically muscles like sphincter muscles, they're smooth muscles, right? Because you don't want to worry about those muscles relaxing when they're not supposed to. And imagine if you have to be contracting it all the time, you're going to have a lot of pain, like sphincters in a lot of places, right? So it's a good idea to keep them in a very good smooth muscle tone. Okay. All right, so they also use aerobic respiration, so they do have mitochondria, right? Because you're only going to get aerobic respiration if you have mitochondrial function, right? So they use ATP, of course. Okay. Now, the mechanism of the contraction. So here you have slow, synchronized contractions. Why is it synchronized? Because they can communicate to each other with those gap junctions, right? So they have electrical coupling. That's called electrical coupling by those gap junctions, okay? Now, some smooth muscle cells, they don't really need activation from the outside, right, to contract. They are self-excitatory. They can contract themselves, okay? So we're not going to get into that type of mechanism, but keep in mind that some smooth muscle cells can depolarize on their own. Okay, so those cells are basically the pacemakers of those types of smooth muscle sheets. So now the mechanism of contraction. Now this is a part that really gets different, and this is a part that sometimes is a bit not very intuitive. Right? So let's pay attention here. So of course you need calcium, right? So you need calcium ions that are going to start the contraction. Now let's look what happens here. So this is from that video. Now this is from another source. This is actually from Lecturio. Don't tell anybody. Trust me, it's a good source. Okay. So here, mechanism of contraction. Okay. As mentioned, it's similar to the smooth to the skeletal muscle because you have those sliding fibers, right? So you do have actin and myosin. They're going to slide, interact with each other here. The trigger of contraction is going to be increasing calcium concentration, calcium level, right? And where is the calcium going to come from? The outside, right? The outside. That is going to bind to calmodulin, okay? Calmodulin. Now, once that activates the calmodulin, now that guy is going to activate myosin kinase. It's not activating myosin, it's activating a myosin kinase. What is a kinase? Enzyme that does what? Puts kinase puts phosphate. You gotta remember. If you don't remember anything out of this class, please remember kinases add phosphates to stuff. And what takes phosphates out? Enzyme has to finish in A's. Takes phosphates out. What's the name of the enzyme? Close to it. Another guess. Phosphor. Come on, people, are you awake? Okay, lollipop for a good answer. Okay, you get a lollipop for stretch. Phosphatase. Yes, you got a lollipop. Phosphatase. Okay, remember, phosphatase cuts down phosphate. Kinase puts on a phosphate. Now look what is happening here. You have here myosin. Here, you have here activation of myosin kinase. Okay, so this is a myosin light chain kinase. So that is telling you the action is on the light chain. The light chain has a kinase power to put a phosphate on the myosin head. Okay, so the light chain is doing the job here. Don't get confused. Okay, so this is activating the light chain. Calmodulin binds calcium, activates the light chain. What is the light chain going to do? Put a phosphate on the myosin head. That's one a very important difference between smooth muscle and skeletal muscle. Okay, so here, activated myosin kinase phosphorylates the myosin head, activating the myosin head. Okay, so we're going to look into more details on what's happening here. Once you activate the myosin head, the myosin is going to bind to the actin and you're going to form the cross bridge, just like you had before with the skeletal muscle. Okay, that is going to use ATP energy, but the whole mechanism of how ATP is used here is different. Okay, so this is not the same mechanism. Okay, how are you going to stop the contraction? Well, we're going to stop it when calcium level goes down, same way as you stop it with your skeletal muscle. Okay, the dephosphorylation of the myosin is going to inactivate it. Okay, so it gets activated by getting a phosphate from the light chain. The light chain is an enzyme now. The light chain is called myosin kinase. There's a part of it that's called myosin kinase. Okay, so here, this light chain, okay, has activity. Myosin kinase is going to put a phosphate on this head and it's going to activate it. So activating binds it, and then when you get rid of the phosphate, then this thing is going to detach. Okay, so let's look at more examples. Okay, so phosphorylation of myosin is the mechanism here. Okay, so here comes your first question. Which protein in smooth muscle has a similar function as troponin does in skeletal muscle? Here is A, calmodulin. Right? Calmodulin does the work in smooth muscle same way as troponin does in the skeletal muscle. And we do have more questions, so keep your iClicker in hand. All right. So here, mechanism of contraction and comparison of smooth to skeletal muscle. First thing you have to do, you got the calcium inside. So calcium has to be sensed by a protein. So here, when you have skeletal muscle, you're going to have troponin that senses the calcium, right? And that does a mechanical job on troponin by changing its configuration or its conformation, right? Now, when you have your smooth muscle here, so let's make sure this thing is labeled. On this side, and here is, right, this side. So here, when calmodulin senses your calcium, it is going to have an enzymatic activation action on calmodulin. So now this the smooth muscle here is going to become active. So here, skeletal muscle, troponin moves tropomyosin, right? And this is a thin filament regulation type of thing. Now here in comparison, you're going to have myosin light chain kinase. Okay, where is the myosin? Here, myosin light chain kinase, that this is going to do the regulation on your myosin. So this is part of your thin fiber. Okay, so now, this one is going to expose binding sites. This one is going to phosphorylate. Okay, different function here. So phosphorylates myosin, that is going to allow those actin myosin interactions. Those are your cross bridges. Okay. Right. So everybody clear with that? Yeah. Okay. I did put up the PDF for this. All right. So here how it's working with the cross bridge formation. Okay. So here, what is happening at first? You have binding sites here for ADP and for phosphate, okay? So your myosin head here does not have that, does not bind ATP directly, okay? So what is happening here? When you have your calcium over there, this is the active site, right? The active site of your thin filament that is exposed here. So this is in the presence of calcium already. So that is going to activate the head here of your myosin, okay? So that is going to allow for phosphorylation of the head. Where is it? It's over here. P1, P1, why is this over there? Okay, this I got a little confused here in a second. Yeah, I should have started this circle with an open circle, not straight into the diagram here. Okay, when your head is attached, you have a phosphorylation of your myosin head. Now the myosin head is going to get rid of the ADP and of the phosphate that is stuck to it. Okay, it's just going to dissociate from it once it is bound to your actin. The dissociation of the phosphate and of the ADP is going to cause the head to do the power stroke. Okay, so this is what is showing here. Okay, the head does the power stroke. Now, After that happens, you're going to have interaction of ATP with the myosin that is going to dissociate that from your actin filament. Okay? So that once that happens, you dissociate from the thin filament, and then here you're going to have phosphorylation of your ATP, which is going to again bring this head back into the cocking position. So ATP is still in the bend position. When it phosphorylates to ADP and PI, it changes the position of your myosin. Now you are able to interact again with your actin filament. It's basically the same thing as your skeletal muscle bridge, cross bridge formation. The difference here is that you have the actin activating the myosin by putting a phosphate there in the first place. And I don't think it shows very clearly here. But keep that difference in mind. Okay. All right, so here, looking at the whole picture of your smooth muscle when it's relaxed in comparison to when it is contract, keep in mind that you have those dense bodies that are holding those filaments together, right? And serving as the anchoring points, okay? And also the dense bodies are crossing with those nets of intermediate filaments. So here is just another also explanation of the mechanism of what is going on with the myosin. Okay. So when you have myosin light chain kinase, that's the one that's going to put the phosphate on your myosin. So that is going to activate your myosin. Now you have another protein that is myosin phosphatase that is going to inhibit your myosin. Okay. So myosin phosphatase inhibits myosin. Myosin kinase activates the myosin. Okay. That's the part that is important to know here. Okay, so that's the activator, that's the inhibitor. All right. Now, remember I mentioned that smooth muscle can make those latch bridge states. What does that mean again? It's going to be, myosin is going to be bound to actin, and it's going to be stuck in there in a contracted form without using energy. So that is avoiding fatigue, and is very efficient for not having a lot of energy usage, right? And is very useful in case you have smooth muscles that have to be contracted for a long time, okay? So your next question is coming up. So do you understand this? Ah, now you woke up. Why is this latch bridge state important for a smooth muscle? Ah, yeah, you contract without tiring, without thinking about it, because it is autonomic nervous system, right? Exactly, that's the whole point. Imagine, I mean, you're in the classroom and then all of a sudden your intestinal muscles get tired and decide to relax. What's going to happen? Disaster. A smelly one too. All right, so here's for your next question. What is the function of the latch bridge state? So the answer is C. Okay, we didn't mention anything about any pH. What is it that? I don't even know what is an pH. I know what is an ADPH. All right. So here we go. Oh, another question. Party. Which of the following enzymes inhibit myosin? So now you have to think about what activates myosin. You think about what inhibits myosin. Myosin phosphatase, right? Kinase activated, phosphatase deactivated. Yes, I did have protein kinase C there, but it has something to do with activation of actin. And where is protein kinase C coming from? Did I mention that? No. That comes later when we get into cell signaling and signal transduction. It gets more complicated, a lot more complicated, okay? But we're not mentioning it here. We're trying to keep it very simple. So we think about myosin kinase and myosin phosphatase. That's it. Where do you find your myosin kinase? Your actin filament. Your actin filament becomes an enzyme that is a myosin kinase. What activates that? Calcium, the presence of calcium that binds to calmodulin. All right, so let's see here. So the smooth muscle fibers are different in organization, right? And in their innervation, that means they can be innervated with nerve fibers in a different way, right? And they're also different in the way that they respond to stimuli, okay? So here, smooth muscles can be placed in two different boxes. One of them is called unitary. The other one is multi-unit. Now that doesn't have to do with the number of smooth muscle cells. It has to do with the number of nerve fibers that are going to be innervating those smooth muscle cells. Okay, so in a single unit or a unitary system, you're going to have one nerve fiber that is going to get there and innervate this whole bunch of muscle cells. Then when you look here at the multi-unit or multi-subunit, right, multi-unit, you're going to have a lot of nerve fibers. They are going to be innervating those smooth muscle cells. So we're going to look into more details on how that's happening, okay? So single unit, unitary, right, is going to need only one muscle fiber that is going to activate those cells, and they are very efficient at transmitting the action potential and the stimuli through all the muscles that are in that patch, because those have a lot of gap junctions, okay? So look what happened here. Unitary smooth muscle, it has one nerve, unitary for the nerve, okay? And those are going to be found usually in the visceral muscles and in hollow organs, okay? So remember how those smooth muscles look when you look at the hollow organs, right? They have those longitudinal and circular fibers, and this we have talked about. Now, what happens here is that your one fiber, it activates, even if it activates only this part of your muscle, it doesn't matter because the gap junctions are able to transmit the signal to the other muscles. Okay. So here, it doesn't need more than one fiber because of those gap junctions. So I have that like 3 or different places, right? So the analogy here is like with Christmas lights. You have all the Christmas lights, they're connected with this wire. And the analogy is the wire is the gap junctions. So you plug one side and all the lights go on. All right. Another question. Okay. Major cellular feature in smooth muscle that contributes to its rhythmicity and ability to participate in peristalsis is the presence of what? And we just looked at it, this unitary thing. Those are your gap junctions. All right. So now looking at this multi-unit smooth muscle, those are innervated by many fibers. Now, one thing that you can find that is different between this and the other one, you don't find those gap junctions here, right? So here you do have the muscle that has a fiber, another muscle has a fiber, but they're not communicating with each other. They're not having the gap junction. Otherwise, it would be, kind of useless to put a lot of fibers if they have the gap junctions to communicate among themselves, okay? So they have very, very few gap junctions. And where do you find these types of multi-unit? You're going to find them in the lungs, large arteries, rector pili muscle, and the iris of the eye, okay? So very few muscle fibers. They are innervated, of course, also by an autonomic nervous system. And what happens here is that you can get those graded contractions. Okay, so you have a little bit, or you can increase the amount of contractions based on the amount of the motor units that are activated. So this involves muscle fiber recruitment. Okay, so you can activate just one of them, or just two of them, or more of them to get more contraction. Okay, so here's what they're found, we just mentioned. Another question. Electrical coupling by gap junctions is present in what? Right, the unitary smooth muscle is the one that can communicate with gap junctions. There is only one nerve going to that. Right. Now, regulation of contraction, right? So many things can regulate the smooth muscle. You can have nerves, hormones, or even local chemicals, local changes can regulate the contraction. So you have in terms of neural regulation, right? You can have graded local or increasing of action potential. You can have a neurotransmitter that gets released. And besides that, as I mentioned, you can also have hormones and local chemicals that can activate the muscle. And of course, if you're activating it by hormones, you don't really need to use a nerve to activate the muscle. If you have a messenger that gets in there and activates the smooth muscle, what's the use for the nerve, right? The messenger is already activating it. And then you have some muscles that can depolarize by themselves, okay? And they do that by a process that involves G protein receptors, which we did not mention it, but keep in mind that's part of signal transduction, okay? And other factors that include also carbon dioxide, pH, and low oxygen can activate smooth muscle contraction.